everyone, to today's conference call. At this time, your lines have been placed on listen only for today's conference until the question and answer portion of our call, at which time you will be prompted to press star one on your touch-tone phone. Please ensure that your line is unmuted and please record your name when prompted so that I may introduce you to ask your question. Our conference is being recorded, and if you have any objections, you may disconnect at this time. I will now turn conference over to our host, Ms. Irene Ahir. Ma'am, you may proceed.
Thank you. Hello, I am Irene Ahir of CDRH's Office of Communication and Education. Welcome to the FDA's 37th in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to today's discussion. Please remember that we are not able to respond to questions about specific submissions that might be under review. Now, I give you Timothy.
Hello, everyone. Welcome again. We look forward to interacting with you today on these important topics and doing everything that we can do as the FDA to greatly expand access to testing so that we can be more and more able to battle this deadly virus. So I did want to start off by reminding everyone that the FDA is allowing tests, diagnostic tests to be used, antigen and molecular tests that have been authorized to be used off-label in testing asymptomatic patients. So that is completely legitimate to do. We're encouraging it because we only have a small number of tests right now that are authorized for asymptomatic screening. So off-label use as directed by the prescriber for prescription tests is completely acceptable and encouraged. In particular, direct antigen tests, that have been authorized. All of them that have been authorized can be used for this purpose. There's rare examples where we've seen asymptomatic data that is not sufficient for authorization, and we would make a very rare and unusual statement in the IFU, in the instructions for use. Otherwise, we just categorically say that all the authorized tests for direct antigen testing can be used off-label by prescription in the asymptomatic population. Again, we don't know the actual performance, and that should be understood by the prescribers that if there's something that's positive, then that's great. But even in that situation, you want to make sure and look at the spread of the virus in that particular area. If the incidence of positive results is low, then it's important to strongly consider a confirmatory test. We would typically say use a high sensitivity molecular test. Hopefully you have arrangements that you can get that result back in 24 to 48 hours. That would be ideal, obviously. Another a different antigen test could potentially be used as well. Whatever you have access to, that's important to be sure and confirm that result. But again, I think high sensitivity molecular test. A different antigen test may have slightly lower performance than a central lab high sensitivity molecular test and could return potentially a false negative on the repeat testing. So all that should be taken into account. But we don't know, unless we've seen data on a particular submission, we don't know the performance in the asymptomatic population. As I said, we are looking and encouraging more and more submissions for asymptomatic claims, for screening claims, and we welcome those. But in the interim, we've made this very clear, and CMS and HHS have made this very clear as well, and have removed any potential roadblocks at the federal level. If there's anything else that we can do to assist in deploying the tests in that matter, if you have questions, please email us at the template email address, and we'll do our very best. Of course, we have FAQs on this. CMS has FAQs on this, as does HHS. I did want to also state that starting in the new year, we will continue these town halls. Look for announcements. We will try something a little bit different in the new year. I want to make sure that those developers of antigen and molecular point of care tests and home tests have the ability to get their questions asked and potentially answered to the best of my ability and our abilities. There's typically a number of callers, and sometimes we don't know if callers don't get their questions answered. So we're looking at different ways of doing this. I anticipate that after we may do this by topic, alternating weeks, but I anticipate that in all cases, by the end of the hour, we will accept all questions. So that I just want to sort of make sure that that every different development group out there gets a chance to ask some key questions, so that we can clarify, explain, stimulate, encourage all test development. So, of course, if you're paying attention to our announcements, we've had more authorizations in the intervening week. Of note is one very important authorization, and that was an antigen, happened to be an antigen test, but it is an antigen test that has been authorized as the first home-based, over-the-counter test. And so we are extremely excited about that here at the agency, and look forward to more and more home testing authorizations, not just for antigen tests, not just for and molecular, but also for serology tests as well. And of course, we had a prior prescription-based molecular test, and we look forward to more authorizations, both by prescription and OTC in the home. Of course, molecular is great if it can be done in the home environment. There is a challenge, though, in that it is more difficult to manufacture molecular tests in quite the same volume as direct antigen, let's say lateral flow tests. And that, and also there may be cost differentials too. But because of the manufacturing capacity of these so-called paper strip tests or lateral flow tests, the authorization yesterday for the first home OTC test was such a paper strip test. The capacity of making those tests is in the potentially tens of millions per month, if not even to the 100 million mark, even from a single manufacturer. So obviously, deploying such testing that has been EUA authorized, by which we've had a chance to look at the test and authorize it and get a good initial feel, at least under the EUA framework, of its performance, so that the public can and users of these tests can have assurances that the FDA believes they are accurate for the intended purpose. So, I mean, that kind of explains why it's so important, because right now there is a desire, I believe, out there to have a lot more testing. There's a willingness to do this, and so we're looking at we're looking at how we can make that testing as widely available as possible, so that consumers even can, and home users can get a fast, accurate result that they can use to make the very best decisions about their lives and those of their loved ones, and other situations that are important for doing that. Okay, we will move into the Q&A portion now, and I look forward to your questions. Thank you.
Thank you. At this time, if you would like to ask a question, please press star 1 on your touch-tone phone. Please ensure that your line is unmuted. Please record your name when prompted so that you may be introduced. Our first question is from Melissa Grisaja. Your line is open.
Hi, good afternoon. Thank you, Dr. Stencil, again for continuing these town halls. They're very much appreciated. I have a question concerning at-home over-the-counter tests going forward, since we now have a baseline with the Illume test that was just authorized yesterday. I noticed that the letter of authorization states that the Illume app is able to automatically report to the relevant public health authorities. And I was just wondering is this a function or feature that you would expect all over-the-counter COVID tests to be able to do going forward, as in have the capability to generate reports of test results and send them to public health authorities? And if that's not the case, and an over-the-counter test did not have a corresponding app, how would you expect this reporting to happen? And then last thing as a side note, the letter of authorization states that the Quick Start Guide and the Illume FAQs are available on the FDA's COVID IVD EUA webpage, and I haven't been able to find them. Right now, there's just a blank spot where the authorized labeling and authorized settings are located. And I wasn't sure if that was on purpose or if it was a glitch, but just wanted to let you know.
Yeah, there's a number of questions. Stay on so I can make sure that I've noted all of them. I'm just making notes right here. So yeah, I noticed the posting was not complete yesterday when I checked it, and I'm not sure why, but we'll double check on that today. It sometimes takes a day or so to get the posting up post authorization. So I will look in that, because I saw the letter of authorization, but I didn't see anything else. So I will check on that, and maybe by the end of the hour I can report back on that situation. In regards to reporting, it's obviously strongly encouraged that developers of all tests consider that, in particular point of care tests and home tests, where it's maybe a little bit more challenging because it's not being done in a moderate or high complexity lab, which has established, or it's easier, at least relatively easier for them. Not that it's easy, relatively more easy for those labs to have reporting features to the public health authorities. It's obviously, critical that the U.S. government has, public health authorities have data on where virus is going up and where a virus is going down. We are constantly looking at effectiveness of programs in testing, and that's just one good measure of how the programs and the amount of testing in an area is being, whether it's being effective or not. So, and the more we have that sort of information in real time, the faster we can make assessments of the importance. And so while it's not what I would call a decision requirement for an EUA, we encourage it. We encourage developers to think about this. We look forward to those developers who don't have one at authorization. I believe the first home test was that situation, and we asked them as part of a post-market commitment to come up with a plan to be able to provide home users, in that case it was RX, and or their prescribers to be able to report that information back. There is a U.S. government apathon that has just concluded. The goal of that apathon is to identify reporting technologies that could be used not just for a specific test, but it's called a design-a-thon. Not just for a particular test, but that's fine for developers to do that, and proprietary reporting is fine. But to find designs and apps and reporting functions that can be used independent of the test performed, so that this eases the desire we have for all developers to make some sort of reporting feature available. If a given, especially a small developer of a test, doesn't have the full capability to really think about this, then having a off-the-shelf app that could be used for them, that would be awesome. So we are pushing that forward, and I don't know all the current up-to-date details on that, but I know we got a lot of submissions, and I know they're moving towards, but they haven't already designated who are the winners in this round of the design-a-thon. Okay, I think that I've answered your questions. All right.
Yeah.
All right.
Thank you so much.
Next question. Mm-hmm.
Next question is from Wendy Strongen. Your line is open, ma'am.
Thank you. I have several questions. Our test is a rapid lateral flow antigen test for home use and for point of care use. In our clinical trial, does the comparator PCR test need to be collected using a nasopharyngeal swab, or is an anterior nasal or mid-terminate swab sufficient? For your application, or what's the sample type again? Was it an anterior nasal swab? It's an anterior nasal swab for the lateral flow test. So does the PCR, the swab for the PCR test, does that need to be a NP swab, or can it be an anterior swab? It can be, as long as the molecular test is authorized for an anterior nasal swab. So we don't, we recommend that you use tests that are used on label. Also, we do recommend that you check with us about the comparator method to make sure that we believe it's a good comparator. We have asked for the comparator to be a high sensitivity molecular test that uses an extraction procedure to concentrate the virus. And then, go ahead. I was just going to say, I believe there's a list that FDA has. Is there a list we can look at to see which ones are recommended, or do you prefer that we directly contact you? I don't believe we've posted a recommended list. We have the results that have been posted already for the RNA reference panel testing. We do use that testing in part to assess whether it's a comparator, and we try to the reference data is important. Some reference data for some tests has not yet been posted, but we have that information, and if you were to want to use that molecular test, there may be no reason not to, but it's not visible to the public what the reference panel results are, so you wouldn't necessarily know. So there could be molecular tests not on that list, for example. We tend to want to look at the more sensitive tests, so you can look at that list and look at the relative analytical sensitivities, and then double check with us. That would be great. Okay.
Okay, thank you. And my other question is about retrospective samples. We want to augment our data using retrospective samples. And the samples that we've been able to obtain, which are purchased remnant samples, are diluted 10 times compared to how we would be actually running them, because these were samples that were put into viral transport medium. So we'd like to be able to take into account that dilutional effect in two ways. We want to limit the retrospective samples to being below a certain PCR value, for example, a CT29. And we'd also like to be able to present a calculated CT value that we believe can be reached based on the fact that there is this dilutional effect.
So yeah, we certainly understand that it's in some ways easier to get these bank samples, and sometimes they're not ideal for, especially for direct antigen tests. We want to use a direct swab. So we are open to the use of those samples, and we understand that it could put your device at a relative disadvantage by using them. We look for and recommend actual data on actual samples. So there have been some direct antigen tests that have included, say, UTM, VTM in their authorizations, at least initially. The first antigen test was that way. And their sensitivity in that substrate was enough for us to go ahead and authorize. So that's our recommendations. If you are going to propose something different than that, I would recommend that you come in and seek, our concurrence before you actually spend money and do the study. We'll endeavor to get back to you as soon as possible, given that point of care tests are a priority. But wherever possible, it is our recommendation that actual direct swabs be used. They can be frozen direct swabs if there are banks out there that have been banking frozen direct swabs for an antigen test, for example, or for a molecular test that wants to use. We will accept that as supplements for fresh samples, but we have some evidence that freezing could actually increase the sensitivity of testing, surprising enough, and we don't want to limit an authorization to any test that says you have to freeze the sample before you test. That wouldn't make any sense. So we do ask for a minimum of 5 positive, for point of care testing at least, 5 positive prospectively collected fresh samples for us to make an authorization decision, and then there's usually a post-market commitment to finish that study. Given that, approximately over a week we have over 200,000 new cases of PCR, of test-positive patients in the United States, we do understand there is challenges in standing up testing studies, but there really is, unfortunately, there shouldn't be any trouble getting the few number of fresh positives that we want to see prior to authorization.
Thank you so much for your question. Moving on to our next question. And as a reminder, please press star one and record your name if you'd like to ask a question. And we do ask that you limit yourself to one question. Our next question will come from Mark Hackman. Your line is open, sir.
Yes, good morning, Tim. One of my questions has been answered. It was regarding the documentation associated with the new antigen test that was approved yesterday. So thanks for checking into that for us. And then my second is just to say thank you very much for bringing out some OTC products, you know, for the public to use. And I'm just wondering, are there a lot more in the pipeline coming down the way? And that's it. Thanks very much.
You're welcome, and you give me an opportunity to mention that there is some editing going on with the IFU for the Illum test. Those edits have arrived back at the FDA, and so that should be posted hopefully pretty soon once they've been authorized. So just keep checking. So obviously, this also displays the flexibility we have in that we have given to companies. I do want to make another late-breaking announcement. I've just been informed that another home test has been authorized, and the authorization has been acknowledged by the sponsor. And I just got text and Skype confirmation of this. So it's not an OTC test, but it is another home antigen test, and the Abbott Binax now has been authorized for home prescription use. So to answer your question, we're very encouraging of both home prescription tests and home OTC tests for Illum. They showed a really good performance in even asymptomatic patients. And we have, we have more developers that I think can meet our recommendations for OTC. We have a substantial interest, and I would expect to see more authorizations. I can't predict exactly when of all sorts of home diagnostic tests, and whether it's prescription and OTC. But certainly, this is FDA flag in the ground that says yes, we are going to authorize OTC tests.
Thank you for your question. Our next question is from Steve Gonzalez. Your line is open, sir.
Yes, hi. Thank you very much for taking my call. I had a question about FDA's policy with regard to reviewing laboratory-developed tests, and if not within 14 days, NCI's assistance with this. A few questions related to that. So first, has FDA implemented this new policy? And second, is there a mechanism by which a lab should submit directly to NCI if it does not hear with an FDA within 14 days? And finally, for labs that were previously informed their LDT would not be reviewed according to FDA's previous prioritization scheme, is there any particular manner in which these labs should resubmit their EUAs to FDA or directly to NCI? Thanks.
Well, we'll start with the last one first. So we welcome emails and questions at our templates email address, and what I can say now is that we are still in discussions with HHS on this topic, and as soon as more details can be given, they will be. Okay, I'll open for another question.
Okay.
Another question in the queue.
Our next question is from Pierre Palachicchio. Your line is open.
Thank you for taking my call. I would like to ask if the actual submission of the tests that have been approved are available to us to read and preen information off of. I think it'd be very valuable for us developers. It would cut through a lot of our red tape to trial and error to send in our pre-EUAs, and it would dot the I's and take care of our development time frame. If those EUA actual submissions that have been approved already, not the ones that are pending approval, are available to developers. Thank you.
Yeah, I'm not an expert on this particular topic. I'll just say that we don't have any plan of posting that information. In almost all cases, if not all cases, there is proprietary information in those applications that is not unlikely to ever be made public, and sponsors, test developers appreciate that. You will understand that in order for us to make the very best assessments, we ask for this kind of this information, and that would include proprietary formulations, so that we can understand. I'll also say that this may be the first time that I'm mentioning this on the town hall call. So we do ask for all developers to provide us as much detail about their test design and designs. And so we do ask for, say, for molecular assays, primer and probe designs. Our office on a monthly basis is looking at the database of mutations, at least that are linked to the U.S. Since we have those primer and probe sequences, our bioinformaticians are scanning the mutation data and flagging anything that could be of concern. We have already initiated reaching out to some developers, and we are, in dialogue with them. If there is something that is existent that would potentially lower the performance of the test, we will work with those developers in every situation and make that information public as soon as possible. We're probably looking at communicating this program more formally in the not too distant future. We are doing this as a service. It's not something that we normally do, but in this pandemic, we feel it's important. We would encourage all developers, of course, when the molecular developer, say, comes in with primer and probe sequences, we are asking for both inclusivity testing, even if and only in silico, as well as cross-reactivity testing, at least in silico for some things. But we do really want test developers post-authorization to be surveying. We would encourage them and recommend to them that they survey mutation status, and for any prevalent mutations that might exist in the population, at least for the U.S., that might affect the performance of their assays. So more to come on that later. We, in our office, have traditionally, even before the pandemic, have made decision summaries available for all of the authorized tests, whether that be a PMA, whether that be 510K or de novo. We do this after it's been scrubbed for confidential information from the developers, to provide just the sort of information that you're asking about that's most relevant. And we believe in our the IFUs and the letter of authorizations and some of them post-market commitments, that we are providing this same sort of clarity to the development community. And if you have questions about designing something or something about your test that even after you've reviewed previous authorizations, you're always welcome to come in to our templates email box and ask those questions, and we'll be as forthcoming about answering your questions as we can. Again, we treat there is a significant amount of company confidential information that we guard and we protect because that's part of our duties, and it's also the law. All right. Thank you, and I think we can go to the next question.
Our next question is from Jessica. Your line is open, ma'am.
My question has been answered. Thank you.
Thank you. Our next question will be from Russell Ivanhoe. Your line is open, sir.
Thank you. I just have a quick clarification, Dr. Stenzel. Something you mentioned on a previous call regarding the prospective fresh samples. You say you recommended a minimum of 5 fresh samples and that you're providing a huge amount of flexibility in allowing the completion of the fresh sample studies post-market. I just wanted to know if that meant that we could submit our EUA and then do those 5 fresh samples in post-market.
For point-of-care studies, home studies, we want to see samples as they would be used in real life. The flexibility that we're allowing is to 5 fresh positives is not meeting our recommended number of fresh positives for point-of-care testing. What the flexibility is that only 5 of the recommended fresh positives and getting fresh negatives is pretty easy. There's no reason not to have fresh positives and fresh negatives in sufficient numbers to meet our recommendations. But it's the positives instead of say our recommendation of 30 fresh positives instead of requiring all those or recommending all of those pre-market for authorization. We're allowing just 5 positives, not just 5 fresh samples. It's 5 positive samples. So we get a read on the performance on fresh samples.
Thank you. Our next question is from Peter Sansanio. Your line is open, sir.
Yes. There's been a number of statements about improving the or compensating for potential sensitivity shortfalls for home testing and or point-of-care testing by serially administering a test. And in the last call, you mentioned, that the 2 tests had to be, in effect, orthogonal, different manufacturers. And my question is that the only serial testing mechanism that one could invoke to address that, potential shortfall, or is there, timing of serial tests of the same, assay that could be used?
As Dr. Shuren and I announced in the Hill piece in September, we are open to tests that fall below the recommended sensitivity if there's mitigations. One mitigation is serial testing. If that's something that you want to pursue, you would do a study with the particular serial testing format that you want to follow with your device. I'm lost a little bit on the orthogonal. I think that was confirmation post-authorization for actual use of an authorized test in low prevalence populations where you might discover an asymptomatic positive and you want to know, is it a true positive? Then we're asking for an orthogonal method. We're recommending an orthogonal method that's post-authorization. That's just normal clinical use. If you were to take a molecular antigen result in a low incidence population, a positive as absolute truth, that potentially could have
untoward consequences. And we're recommending, the CDC recommends that you test with an orthogonal. But pre-market for authorization purposes, the serial testing suggestion recommendation is one way to mitigate a test that on a single test, say, single test result in a given person may not meet the recommended bars that we have for authorization. And by testing on a set schedule, you know, say it's every day, every other day, every day, once, day 1, day 3, day 1, day 4, day 1, day 5, depending on whatever serial testing program you want to have for your device. It would be with your device. To bring that sensitivity level up to our recommended level, we're totally open with that. There are potential other mitigations. You mentioned orthogonal tests. That could be useful. The test, that orthogonal method has to be authorized for the setting in which you are going to deploy that. And we would want you to come in with data that shows that whatever testing scheme, serial testing scheme, orthogonal testing scheme you want to use in order to mitigate a lower sensitivity. We're open to that. We just want to see the data, and we would authorize based on the data and sort of the study design that we use. So if you came in and you say, we tested with our test, 2 tests, once on day 1, once on day 3, combine the sensitivity at our threshold, recommended threshold for sensitivity for a point of care or a home test, then that's how we would authorize it. We would say, okay, this test is authorized if you perform it on day 1 and day 3, and this is how you interpret the results. So hopefully that's helpful.
Thank you. Our next question is from Richard Darren. Your line is open.
Hello. I wanted to ask you more of a policy question on compliance standards. Is the CDRH still requiring full compliance with ISO 13485, and will it eventually require compliance with the IVDR?
So we are currently, obviously, there's public plans to merge our 820 quality system requirements with 1345. That, to my knowledge, hasn't been implemented yet. We're heading in that direction. That has been publicly announced. Can't say exactly when that would be. We're obviously in discussions with worldwide regulatory authorities, including Europe and in Asia and all parts of the world who want to participate in those dialogues. I think our center has been very clear and forward-thinking that we would love to see worldwide harmonization around quality system requirements. Our willingness to conform and move towards 1345 signals that. Also the single audit program, the single review program, which has not been stood up yet, but we're obviously, it's publicly known that we're very in favor of that. Let me be clear. So we're talking about non-EUA situations here, okay, on full compliance with quality system regulations, which currently for the FDA are 820. In the EUA, we have waived most of those quality system requirements, and those that still need to be adhered to are highlighted in our various communications and in the letters of authorization. So we're under an emergency use authorization situation, we're waiving the vast majority. We've seen a huge influx of developers, which we welcome, who have never submitted anything to the FDA before. And obviously, it's not going to be an overnight situation where they're going to be
fully compliant with A20. That's the current situation anyway. When it comes time for us to transition, once say the emergency is no longer declared, and there's going to be a time of period which we're planning to issue a guidance around that transition period and the amount of time to convert EUA applications to full authorizations. And that would also, in all likelihood, include recommendations about how to move from what we allow under an EUA situation for quality systems into a time period, a timeline to become compliant with at the time that occurs with whatever quality system that we are recognizing and asking for. So hopefully I addressed that question and also made crystal clear about the differences between non-EUA and EUA situations.
Our next question is from Louis Ferland. Your line is open, sir.
Yes, hello. Thank you for taking my call. My question is a follow-up to a previous one that we had earlier today, where the developer indicated that she had an assay she was developing that was to be carried out on anterior nasal nares samples, and she was asking if the comparator would have to be done with NP swabs or if doing this with anterior nasal nares samples would have been okay. I'm a little bit puzzled by your answer that it would have to be done with a test that is authorized on whatever sample they choose to use for that comparator assay, because the template indicates that categories of anatomical sites divide between upper respiratory and lower respiratory and others, such as saliva, was such that NP swab was a representative for NP, OP, midturbinate, and anterior nares. So if we follow that, it would seem that any assay that is authorized for NP would be allowed for nasal nares, would be authorized for nasal nares, or did I understand this incorrectly?
I think the question earlier was a little bit different. They were asking if they could use an anterior nasal sample for the comparator test. It's just much more well tolerated, and they can enroll patients more easily than if they were to require an NP swab. Of course, if somebody, a developer, wants to use an NP swab for the comparator test and an anterior nasal swab for their test, that's fine. I would caution that could be putting your test at a disadvantage, because the NP swab may be more sensitive than the anterior nasal swab. If you want to use a comparator that is only authorized for an NP swab, and you want to use that comparator, then whatever anatomic site you're looking for, as far as doing the clinical study, you're fine to use the NP swab. I mean, that's the preferred swab for all of our work. I was just giving flexibility to this developer and other developers if their molecular comparator is authorized for anterior nasal and NP, they can choose whatever authorized swab for the comparator testing that they want. But if a test is coming in only for NP swab, which is probably, unlikely these days, in order to show the best concordance, you want to probably use the same anatomic sites. You don't want to do NP to NP. These are just practicalities that go beyond our specific recommendations and go into, what is a fair comparator for your test? What's going to put your test in the best possible allowable light? I'm looking to authorize tests. I'm not looking to deny tests, and I'm giving the feedback to be able to give you and other developers the best feedback to make that the most likely possibility. There is one subset of tests where we are asking for either an NP swab or a mid-turbinate swab, and again, the comparator test has to be authorized for one or the other of that. And that is for saliva or oral fluid. We still are asking and recommending that the comparator swab be at least a mid-turbinate swab, which is much more tolerable than an NP swab. Hopefully I clarified that.
Our next question is from Kathleen Kennedy. Your line is open, ma'am.
Hello. Thank you for taking my call. I was wondering if we are submitting an EUA for an antigen test for lab use only, do we need to include the 5 fresh samples that you've been talking about this week and last week?
Okay, antigen test, initial EUA submission. Can you include, can you just submit it with 5 fresh positives and not the full 30 or so?
No.
Positive, whatever. Go ahead, clarify.
One moment. Let me open her line back up. One moment.
Hopefully he's still there.
I am.
Your line's reopened, ma'am. Go ahead.
Hello. We have all the clinical.
Please clarify. Yeah, go ahead. I'm sorry, I'm having a hard time hearing this caller. Am I being heard?
Unfortunately, she just disconnected.
Okay, please call back in and ask your question. We'll try to sneak you in. If not, please just email the template email address asking that Dr. Stenzel, Tim Stenzel, I go by Tim, gets looped into making sure that you get a response. Sorry about that disconnect. I guess we can take the next caller if there is a next caller.
It's from Wendy Cho. Your line is open.
Thank you for taking the call. Hello? Can you hear me?
Yes. Yeah, I can hear you.
Yes. Yeah, so I just want to clarify one thing. So we know the oropharyngeal, which is throat swabs, that kind of, we can use that once we validate the NP swab and belong to one category. So I have a question about the oral swab. Does oral swab belong to saliva category, or how does that work? Because we validated the saliva test.
Yeah, so there's a difference between an oral swab that's collecting saliva or oral fluid, and between that and an oral pharyngeal swab, which is in the back of the throat.
Right.
Okay, so if you're going for collecting oral fluid, then that's oral fluid or saliva, and more and more developers are seeing the benefit, and we see the benefit of if it's saliva to make sure it's not a coughed up lower respiratory sample, but just saliva, and there's different methods just to collect that. And so we urge that. But so if you're talking about an oropharyngeal sample that doesn't need an NP comparator, I would still say that an anterior nasal swab or a mid-turbinate swab may be better received than an oropharyngeal swab. And for home use, we're recommending against the use of oral pharyngeal swabs, and obviously against the use of NP swabs in the home situation. Mid-turbinate swabs can be used if they're intended to be used in the pediatric population. We're recommending a safety feature on the mid-turbinate swabs so that they're safe to use in kids. And obviously, if the test is going to be authorized for all ages, or at least down to a certain age, we want to see data. We want to see that the study does perform testing on kids. Okay.
Yeah, so for oral swab, we will treat it as saliva, that category, if we don't do the oral-free gel, just the oral swab.
Yeah, then we would want to see a mid-turbinate or a nasopharyngeal comparator in that situation.
Okay, so even if we already validated the saliva, which will still validate the oral swab for that.
Well, that might be something to come into, but in general, we don't. The best comparator. Sorry for the phone in the background. The best comparator is for any oral fluid or saliva is an NP swab or a nasopharyngeal swab.
Okay.
I'm saying a nasopharyngeal or a mid-turbinate swab. Sorry.
Yeah. So we did validate our saliva with the NP swab. So we just want to see if we can use the oral swab.
Yeah, we're not recommending that saliva be used or oral fluid be used as a comparator for the opposite. We're not recommending that. We're recommending a mid-turbinate or a nasopharyngeal, even if you already have saliva authorized. Mm-hmm. We've seen some quite variable data with oral fluid and saliva, and so for those particular claims, for those that come in for EUA authorization, that's what we're asking for.
So even the LDT is okay? So if we use the oral swab for LDT?
So, my focus is directing right now to recommendations for
manufacturing
tests that come in for EUA authorization. Which at the moment is until we finish our discussions with HHS. It's our recommendation. I mean, certainly other test developers can take and use our recommendations, but these recommendations for now are largely directed towards, in fact, completely towards kit developers.
Kathleen Kennedy has returned to ask her question. Your line is open, ma'am.
Hello, thank you. My question is, if we're submitting, my question is, if we're submitting an EUA for an antigen test for lab use only, and we have everything that's recommended in the template, do we now also need to include 5 fresh samples?
So you're submitting a moderate or high complexity test, not a point of care.
Correct.
That's a little bit unusual situation. For molecular tests, we do allow frozen bank samples, and we haven't been asking for fresh samples always. But that kind of changes too. So I'd look to the template. But in this particular question, about antigen, I think it's best to approach your reviewer. If you don't have a reviewer yet, the template's email a box. If you haven't submitted yet, the template's email a box to ask that specific question. In fact, I just got a Skype from our lead antigen person saying yes, direct her towards the template's email box, and we will address your question as quickly as we can.
Okay, thank you very much. Thanks for getting.
We have a follow-up question from Richard Darren. Your line is open.
Yes, with regard to EUA and compliance, can you specify what quality system provisions will be required for non-EUA tests versus EUA tests?
For non-EUA, it's our standard recommendations for quality system and compliance activities. It's only for EUA tests that we are offering standardly. I will give a caveat to that. To it's only typically for the lion's share situation we're offering for EUAs. Most of the requirements are waived. There are situations where non-EUA tests and testing are impacted by the pandemic. For example, shortages of certain key supplies, plastics, tips, tubes, plates, where a given manufacturer of testing outside of an EUA, so there's platforms used for both EUA testing and non-EUA testing. We are requesting basically, a pre-sub, a Q-sub for those situations, and we will take a look at that. We have also waived some of the requirements for, say, non-SARS respiratory panels and tests and we have guidance out on that, immediate effect guidance, that waives some of the quality system requirements. Not all of them, very specific elements. So it's going to be very specific direct feedback on particular issues. So for our guidance for, say, VTM already, we have immediately effects guidance for VTM, immediately effect guidance for various consumables for non-SARS respiratory tests, for example. Look to those guidances for where we're giving the ability to not follow everything under the quality system, but it's very specific categories. So, and then if there's any questions about those or anything else that's not covered in by an immediately effect guidance, do come to us. You can use our template email address for that as well, or you can go to whoever was your branch chief or reviewer for your non-COVID submission.
Thank you. This does conclude the question and answer portion of our call. I will turn our call back over to our host, Ms. Irene Ihir.
Thank you. This is Irene Ihir. We appreciate your participation and thoughtful questions. Today's presentation and transcripts will be made available on the CDRH Learn webpage at